Hello everyone, welcome to my channel Anupma Biology classes. In this video we will see classification of fungi part 2nd, and here we will discuss about the division Ascomycotina. Before this, in the previous video we discussed about the two divisions of fungi. First one is the Mastigomycotina and the second is Zygomycotina. So in this video we will discuss the third division, which is Ascomycotina. This video is taken from CBSE class 11th, part 11th of chapter 2nd. Now let's start the topic. So in division Ascomycotina, which belongs to class Ascomycetes and also known as the sac fungi. So the main characteristic features of Ascomycetes are as follow. Ascomycetes are a large group with over 30,000 species, include diverse types such as brown, green, blue and pink molds, powdery molds, cup fungi, yeast, morels and truffles. Most of them are terrestrial and occur as saprotrophs or as parasite. The parasitic forms are responsible for several diseases in plants, like powdery mildews, ergots, etc. The discovery of the sexual stage, which is known as the perfect stage of the some fungi imperfectly, that is Deuteromycetes, has shown that they belongs to Ascomycetes. The yeasts include forms that are unicellular and are among the simplest of the sac fungi. Except yeast, all Ascomycetes are mycelial. The mycelium is well developed and branched. The hyphae are septate and multicellular. The septa are characterized by the presence of simple septal pores. Each septum possesses single minute central pore through which protoplasmic strands pass from one cell to another. These pores also show plugging of different types. The Ascomycetes are characterized by complete absence of motile structure in the life cycle. Yeast normally reproduce asexually by budding and fission. So this is budding and this is fission. Oidia stage, similar to yeast, is found in some cases. Majority of Ascomycetes reproduce asexually by the formation of conidia, which is singly known as conidium or conidiospores. These are non-motile mitospores produced exogenously, mostly in chains from the tips and sides of hyphae. Here in this picture you can see this is the conidiophores. They may have distinctive colors like brown, green, blue or pink, which gives the fungi their characteristic appearance. The greenish or bluish growth on bread, some fruits or old leather is due to members of Ascomycetes is known as examples like the Penicillium. This is the green, and the second is Aspergillus. Conidia are born on special hyphae called conidiophores. The conidiophores may occur scattered like Spherotheca and Erisife, or aggregated to form some peculiar structures like Acervulus. Here in this picture you can see this is the Acervulus, Cynema, this is the structure of Cynema, Sporodochium and Pycnidia. This is the Pycnidia. Sexual fusion occurs by gametic copulation, that is shown in the yeast, gametangial contact, that is shown in Pyronyma, spermatization which is shown in Ascobolus carbonarius, and somatogamy, which is seen in Pezaiza. So here sexual reproduction takes place in 3 stages. What are the 3 stages? These are plasmogamy, karyogamy and meiosis. Plasmogamy results in bringing together the two compatible nuclei in a dikaryon. It is not immediately followed by karyogamy. The intervening phase between the plasma and karyogamy is called dikaryophase. The cells of this phase are called dikaryotic cells. Then karyogamy, which forms the diploid stage, then the meiosis occurs. Some specialized dikaryotic cells of dikaryophase functions as Ascus mother cells. In this cell, the two nuclei of opposite mating types fuse by the karyogamy to form a diploid nucleus. The resulting diploid nucleus then divides by meiosis to form and then mitosis to form 8 haploid nuclei, which results in the formation of 8 haploid ascospores. This sexually produced ascospores remains inside the sac-like structure called ascus. Half the number of ascospores in an ascus belong to one mating type and remaining half belong to second mating type. The asci are club-shaped, elongated, ovoid, globose or rectangular in shape. The ascospores are liberated from ascus and germinate to produce new mycelia. The asci in most of the Ascomycetes are produced inside the complex fruiting bodies called Ascocarps. However, in some cases, the Ascocarps are not formed as the asci are produced naked and singly, like in case of yeast. The Ascocarps are of three types. First one is the apothecium. Here you can see the structure. This is the apothecium. In plural form is apothecia. These are cup-shaped or disc-shaped fruiting bodies. The club-shaped asci are intermingled with sterile paraphyses in the upper fertile layer called hymenium. Just below the hymenium is subhymenium and fleshy part hypothecium. In this picture you can see examples like Pejija, Morchella and Ascobolus. After this, the second type of Ascocarp is Perithecium. In plural, we know that perithecia. It is a flask-shaped fruiting body which has a definite apical pore called ostiole. Example is Claviceps. And then the last one is known as Clistothecium. In plural, we know as the Clistothecia. It is a small spherical or ovoid indehiscent fruiting body which is completely closed from all the sides. Examples like Eresipi, Saprotheca. The ascocarps of some Ascomycetes are edible like morels, truffles, etc. So see here, there are some examples before this types of apothecium. So Clistothecium, perithecium, apothecium and pseudothecium. And examples of Ascomycetes, first one is the yeast. Yeasts are a group of about 18 genera which are characterized by the following structures like they are unicellular and non-mycelial or pseudomycelial. They grow in sugar substratum. They reproduce asexually by budding or fission or both. Depending upon their mode of asexual reproduction, the yeasts are characterized into three types. First one is the budding yeast, which is known as Saccharomyces. Then fission occur, fission yeast, which is known as here Schizosaccharomyces, and then halobial yeast. Both budding and fission both occur in this case. This is Saccharomycoids. The diploid zygote functions directly as ascus and produce 4 to 8 ascospores. The yeast in which the ascus formed is reported are called true yeast. In some cases, The ascus formation is not known, but they resemble yeast. Such forms are called false yeast, like Candida, Mycoderma, and included in Deuteromycetes. Most of the yeast have ability to ferment certain sugars. Now, the second example is Aspergillus. The fungus is commonly called black smoky mold. Most of the species are saprotrophic and grow on dead organic material like dung, wood, bread, butter, suitcases, leather boots, jams, syrups, and majority of other substratum. The fungus most commonly contaminates laboratory cultures and hence are called weed of laboratory. It causes rotting of apples, pomegranates, figs, cigars, and tobaccos, onion, etc. A few species are parasite and causes disease Aspergillus fumigatus called pulmonary aspergillosis and ear infection. Aspergillus flavus contaminates the dry foodstuffs such as ground nuts, areca nuts, etc. The fungus produces aflatoxins which are carcinogenic and may cause cancer in human beings. The genus is microbically important as a few species of these genus are used in fermentation industries. For example, Aspergillus oryzae is used in the preparation of an alcoholic beverage that is sake in Japan. Aspergillus niger, which is used in citric acid and gluconic acid fermentation. Aspergillus terus in itaconic acid fermentation. Aspergillus flavus oryzae and Aspergillus tamari in cozy acid production, and some species are used in the production of antibiotics, aspergillin, vitamins, enzymes, and the preparation of cheese. The fungus Mycelium consists of branched septate interwoven hyphae which ramify over the surface of substratum. Asexual reproduction occurs by the formation of conidia on special branches called conidiophores. Each conidiophore arises from T-shaped foot cell and terminates into a solen vesicle. The vesicle produces a large number of finger-like projections which is known as asterigmata. Here you can see this is the asterigmata, finger-like projection, which grow in all directions and bear chain of conidia. They are variously colored, characteristics of their species. Now the next example is Penicillium. The genus is commonly called green mold. Most of the species are saprotrophic and grow on decaying vegetables, fruits, meat, and a great variety of rotting plant and animal remains. A few species are parasitic and causes disease. Penicillium chrysogenum is used for the production of antibiotic penicillin. Earlier, the drug was obtained from Penicillium notatum, another species which is known as Aspergillus grysophilum is used to produce antifungal drug Grysophulvin. Penicillium roqueforti and Penicillium camemberti hydrolyzes casein of butter and fat and impart characteristics flavor to cheese. Certain organic acids like gallic acid, fumaric acid, and citric acid and enzymes are obtained commercially from different species of Penicillium. Penicillium italicum, which causes blue mold, and Penicillium digitatum causes green mold of the citrus fruit in storage. A few species causes a respiratory disease, penicillosis, in animals and human beings. The fungus is mycelial and consists of branched septate interwoven hyphae which ramify over the substratum or deeply penetrate it. Asexual reproduction occurs by the formation of conidia or special hyphal branch called conidiophores. The terminal end of conidiophore has broom or brush-like appearance, technically known as Penicillius. After this, the next example is Neurospora. The fungus is mostly saprotrophic. The species of Neurospora grow as reddish superficial growth over the substratum and therefore commonly called pink bread mold. Many species of Neurospora have been used as convenient organisms for genetical and biochemical studies. The most important discovery that the genes act through enzymes and one gene one enzyme theory was first put forward by Biddle and Tatum through their experiments on Neurospora. Next is Claviceps. The most common species of the genus is Claviceps purpurea, which causes ergot of rye, that is sickle cereal. The disease also appears in wheat, barley, and oat. The grains of diseased plant are replaced by hard horn-shaped purplish sclerotia or ergot. Eating of infected cereals causes ergotism. The disease results convulsions, paralysis, hallucination, and final death of man. Consumption of ergot also causes several diseases in domestic animals. It is of two types, gangrenous and spasmodic. The sclerotia of Claviceps contains several alkaloids called ergot alkaloids. The most common important alkaloid is lysergic acid. So, it is prepared. See here, this is gangrenous type of disease and this is spasmodic. So the most important alkaloid, which one is lysergic acid? It is prepared through fermentation activity of Claviceps spelli. The other alkaloids are derived from it. The drugs LSD is D-lysergic acid dimethylamide 15. Certain ergot alkaloids are also used in medicines. The drug ergotine, a mixture of alkaloids, is widely used for the cure of uterine troubles, hemorrhages, and childbirth, migraine, and enlarged prostate glands. After this, the next example is morels. A species of Morcella are commonly known as morels, sponge mushrooms, or gucci. The fungus is saprotrophic and grows in deciduous forest on humus soil. The visible part of the fungus is fruiting body, Ascocarp of the apothecia type. Each Ascocarp has a fleshy, sponge-like, conical dark brown colored pilus or head and a stalk-like tip. It is edible fungus and considered as a table delicacy in India. It is grown in India, especially in Kashmir and Punjab. Now the last example is truffles. They are edible Ascomycetes having tuber-like subterranean fruiting bodies called Ascocarp. The Ascocarps are dug out with the help of trained dogs and pigs. And the examples are tuber uncinatum and tuber estivum. So this is the end of video in which we discuss about the Ascomycetes or Ascomycotina division. The last video will be on the Basidiomycetes and Deuteromycetes. So this is the end of video. If you understand this video, please like and share it and subscribe to my channel Anupma Biology Classes. If you have any questions, any queries or any suggestions, you can ask in the comment section below. Thank you for the watching.